Yeah, thanks, Amber. So as Amber have mentioned, I'm Emily. I work as a bioinformatics system analyst on the PIC-SURE team, so specifically the Biodata Catalyst powered by PIC-SURE platform. And so today I'll be introducing some of the data that's on BDC, as well as how to find that data on the PIC-SURE platform. So here's just a quick rough breakdown of some of the data sets that we have available in BDC, broken up by phenotype or focus. And so you can see that majority of the data sets are heart, lung, blood, and sleep related, which makes sense since this is the National Heart, Lung, Blood Institute Biodata Catalyst project. And so just to give you kind of an overview here, we've got several COVID-19 data sets, some sickle cell related data sets, as well as some multi-phenotype data sets. So whatever you're interested in, we probably have something related to it. Now, to give you an idea of the magnitude of data available on BDC, so we have over 3.42 petabytes of data available to you. So that consists of over 280,000 participants, over 490,000 data files, and over 150,000 whole genomes. So it's quite a bit of data that is available on Biodata Catalyst. And as you can see here as well, a significant portion of the data we have on BDC is part of the TopMed initiative, which is the Transomics for Precision Medicine consortium or initiative. And so you can kind of see the breakdown of these data sets as well by ancestry and ethnicity. So there's a variety of different ancestry ethnicity, if that's something of interest to you in your research. As well, again, as the breakdown by phenotype. So you can see here, again, we've got blood, heart, lung, and multi-phenotype, as well as some sleep-related data sets there. So just kind of a sneak peek at some of the data that we have available. And so that being said, even though that's what we have available in the ecosystem today, Biodata Catalyst is always ingesting new data. And so you can check the Biodata Catalyst website for a full list of studies available on the ecosystem. So here we've provided the link for you. So you go to the Resources tab and click Data, and then you're able to click Explore Studies to get a full list of the studies that are available there. So if you're looking for one in particular, this would be a good place to start. So that was just a brief rundown of some of the data we have. I didn't want to get in too deep because I'm having a feeling with these next couple of topics, we'll be diving in deeper to some of the nitty-gritty of what we have available. So now I'm going to tell you about PIC-SURE on Biodata Catalyst. So PIC-SURE really empowers researchers to access data, and it facilitates approachable research for all skill levels. And so you can, what PIC-SURE allows you to do is search at the variable value and genomic variant level. So you can search for variables of interest. You can apply filters onto these variables to create a cohort or select certain participants. And by doing those two tasks, you're able to create a data frame that's ready for research or analysis without opening any files or mapping to data dictionaries. So we kind of do all of that file ingestion for you, so that way you can really get jump started on your research. So what is PIC-SURE? PIC-SURE stands for Patient Information Commons Standard Unification of Research Elements. And so as I had mentioned, it allows for searching and exporting at the variable and variant level. It integrates the clinical and genomic data sets across BDC. And there are two main parts to PIC-SURE. There's the UI, or the user interface, that allows users to search available data using queries to build cohorts. So that's the point and click interface that I'll be demonstrating later. And then the results can be exported via the API, so the application programming interface for analysis. So that's using a programming language such as Python or R. So I'll be briefly showing you that as well. And so as I had mentioned earlier, PIC-SURE really allows you to get jump-started on your research because it means that you don't have to spend time accessing the files, opening the files, decoding the data dictionary, and using that information to parse the files and the tables and variables in order to eventually select the participants or apply your inclusion-exclusion criteria to create your data frame and analyze the data. What we do is we do that for you, so that way you're able to just apply the filters in a really easily usable interface, and then you can create your cohort, create your data frame, and just get started on your research. It's all streamlined for you. So before diving into the demo, I did want to mention that there are two main access points of PIC-SURE. There's PIC-SURE Open Access and PIC-SURE Authorized Access. So let me tell you a bit about how they're different. PIC-SURE Open Access allows any user with an ERA Commons ID to search any clinical variable in PIC-SURE. So if you're familiar with Biodata Catalyst data, we use dbGaP authorization, so that is the database of genotypes and phenotypes. So in order to access participant level data, you are required to have an authorization from dbGaP to access that data. However, with PIC-SURE Open Access, you don't need that. So you can search any of the data that's available on PIC-SURE without authorization to access that. So it's mainly used for data discovery or figuring out which data sets you want to request access to. Now, on the other hand, in PIC-SURE Authorized Access, that allows you to access the participant level data of the data you are authorized to access. So after you're granted access to the data, you're able to go here and actually export the data and start your analysis. So as I had mentioned, for access authorization, no approval is required with open access. You just need to log in with your ERA Commons credentials. However, on the authorized access, you do need dbGaP authorization. For data types available in open access, you're able to view the destigmatized clinical variables. And so what this means is that the PIC-SURE team goes through the data and removes some potentially sensitive information in order to protect or contribute to the patient confidentiality agreements that we have with the participants of these studies. However, with authorized access, you're able to access all phenotypic and genomic data that you are authorized to access, so there's no limit there. Additionally, as I mentioned, with authorized access, you get access to the participant level data. However, with open access, you just get the aggregate counts. So I'll be displaying this a little bit more so we can get an idea of how this can be useful. All right, so I think instead of going through these slides, I wanted to put these slides in so that they're available to you, but I think I will actually start by doing a live demo and showing you each of these pieces of PIC-SURE. So here I've navigated to the PIC-SURE website, and so I can click Login with ERA Commons. And so now I am logged into PIC-SURE. And so this is the landing page of Biodata Catalyst powered by PIC-SURE. And so here you can see that we've explained to you what the difference between authorized access and open access is, in case you're still unclear of what that distinction is. And as we can see, based on my login here, I can access 32 studies, or I've been granted access to 32 studies. However, in open access, I could query from all 65 studies that are currently available in PIC-SURE. So you can also see the participant count difference here. Now, if I'm interested to see what data sets are available on PIC-SURE, I can scroll down here to the data access table. And so here you can see that we've listed each study based on study and consent group. And you can get some information about this. So we've included the study focus, study design, as well as some count information such as the number of clinical variables and number of participants. Additionally, what is helpful here is I can see which ones I've been granted access to and which ones I am not authorized to access. So these two studies, the active 4A and 4B COVID-19 studies, I do not have access to yet, but I can see this request button, which will actually take me to the dbGaP page of the study. And so here's where I would actually proceed with submitting a data access request through dbGaP if I'm interested in this study. However, I can see that this Amish study or the RX study, I have been granted access to this. So that means I can access that participant level data. So let's go ahead and build a query in open access here. So I can go to the open access tab of PIC-SURE. And here you can see this search bar, and that's where I'll search a variable or a term of interest. But here, let's look at the results panel quickly. So here I can see that I can query from over 286,000 participants. And I see this breakdown by study and consent group. So over here, we can see that 15,000 of those are in the FHS or Framingham Heart Study cohort, and some other ones as well. So GenSalt, HCHS Soul. So this really gives you an idea of, kind of performing a power calculation or a feasibility study of which numbers of participants are in which studies and which would make sense for me to request access to. So let's try some things, shall we? So let's say I'm interested in smoking variables. So I can search smoke. So I typed in smoke to the search bar and hit enter. And this returns all variables that are in PIC-SURE that have a match for smoke. So we can see here that there's over 1,000 variables that match this search. So we can use these tags over here to filter this a little bit. So let's say I'm interested in the DCC harmonized data set. I can add that tag. So let me do that again. If I hover over it, I can either exclude results from that study, or I can include only the results from that study. So let's include only the results from the DCC harmonized data set. So you can see that we filtered down to the DCC harmonized data set and can only see variables from that study. And so the DCC harmonized data set, the TopMed group organized the Data Coordinating Center, the DCC, and the curation team undertook this effort to harmonize many different TopMed studies. And so the DCC harmonized data set here will actually query across multiple TopMed studies. So it's a really useful tool if you're interested in TopMed data sets. I would recommend using open access, the DCC harmonized data set to discover some data that way. So here I can see that current smoker baseline 1 indicates whether the subject currently smokes cigarettes. Let's say that that's one that I'm interested in. So I can click on this filter button. And we can see that since this is a categorical variable, so this variable is broken into different categories, I can select or filter to only or both, only one or both of these values if I'm interested. So if I only want currently smoked cigarettes, I can check the box there and it moves over to this selected values portion. But let's do both actually. So let's do all of the people that do not currently smoke cigarettes and all of the people that do currently smoke cigarettes. So once I've selected those, I can click Add Filter to Query. So in our results panel over here, you can see that the total number of participants has updated to reflect my query criteria. So there's over 211,000 participants that have either currently smoke or do not currently smoke cigarettes in the DCC harmonized data set. And again, we can see that breakdown by study. So since there's over 15,000 participants with information from the Framingham cohort, maybe that would be a good study for me to request access to. Let's try a different variable such as BMI, so body mass index. So I have searched for my search term BMI, and we can see that the DCC harmonized dataset tag is still in place. And so I'm only seeing results from that study. So here, body mass index calculated at baseline looks like a promising variable for my search. So I can click filter. And here we see that this looks a little bit different now because this is actually a continuous variable instead of a categorical variable. And so to help you, apply a meaningful filter, we've displayed both the minimum and the maximum of this data. So that way you get an idea of what range exists in the data. So we can see that there's a minimum value of about 11 and a maximum value of about 92. So let's add let's say a minimum BMI of 30, and we'll leave the max blank just to have that be the default maximum. So if I click Add Filter to Query, Again, we can see that this filter, my BMI filter, has been added to include only participants with values greater than 30. And I'm still filtering on my smoking criteria. So I've got currently smokes or currently does not smoke and the BMI filter. And so there are over 65,000 participants that meet this query criteria. So I think that's a quick rundown of open access. Let me pause quickly to see if there are any questions before flipping over to authorized access to show you that tool. Okay, great. And so, like I said, you don't need any authorization to access data to use this tool. That you could follow, as long as you have an ERA Commons credentials or an ERA Commons account, you're able to log into PIC-SURE and do these queries with me. You can get the same exact results and discover data. And so, however, with the authorized access, you will need to have authorization to access at least one data set in order to use this tool. And so depending on your authorization, your results may look different than mine do right now. So you can see I've switched over to authorized access, and a few things have changed here. So you can see here that the data summary in the top of the results panel is no longer obfuscated. So in the open access, it was plus or minus 3. Now I can get the official number of participants because I have access to this data. Additionally, I can see the number of variables that are included in my cohort. And I also see a few tools in the tool suite. So I can select and package data or look at the variable distributions of my cohort. So by clicking that here, I can get a sense of what my data currently consists of. So we can see here that there are a lot more people that do not currently smoke cigarettes in my cohort that I've selected than currently do smoke cigarettes. So maybe that's something to consider as I proceed with my research question or analysis. Additionally, if you're interested, these are somewhat interactive, so you can actually zoom in on different parts of the graph, which is also helpful. All right, so let's just add, or let me show you one more thing that is useful to, or using PIC-SURE. So let's say that I'm a researcher and I'm interested in the age of onset of asthma or the age at which asthma started in participants. Now that variable can look very different across many different data sets. Maybe sometimes it's called age asthma started, or what age were you first diagnosed with asthma, or the like. So it can be really hard to search for terms or variables that are related to that. So one way to do this is to search asthma, and we can see there are over 700 variables. So if I scroll down, I can actually use the variable tags to help filter this a bit. So we can see here, there's the age tag. So I can include the age tag. And now we can see all of the variable results that have to do with asthma and age. And so here I can just quickly read through and I see asthma at about what age did asthma start. I could either filter on this variable or I can simply add it to be exported. So by clicking this adding to be exported, it adds the variable to my data frame. And that way it kind of allows for like a quick start to harmonization across studies. So this can be a really useful tool if that's something you're interested in assessing multiple studies. So I can just quickly click through here, add some of these, age diagnosed with asthma, and things like that. All right, so I've got my cohort. I'm feeling pretty good about the data I've selected. So let's go ahead and select and package the data. So here we can see a quick summary of my data set. So I've got over 63,000 participants and 16 variables. And I see that, PIC-SURE is telling me, hey, you've selected a lot of data. Let's remove some because this is too much to package. So I can remove these two variables and kind of review the variables that I've selected or filtered on. And so when I'm ready, I can click Package Data. And so while I'm doing that, I'm actually going to show you that I'm planning to export this data into 7 bridges using the PIC-SURE API. And so I've actually already created a project with Dave here called the Picture and Data Browser Demo. And we can see here that I have a picture export analysis. And so switching back over to my picture UI here, I can see once it's ready to be packaged, there's an export to 7bridges button. So if I click that, it displays all of the information and all of the steps I need to take in order to get my data into 7bridges. And so I can click this button to go to the public project in 7bridges, or I can just copy the query ID into a project I've already created, which I've done here. And so here, this is publicly available code. So the picture team has created these step-by-step coding examples to show you or show you how to accomplish certain tasks with the picture API. So it breaks down exactly what you need, which steps you need to take. And we've actually provided this example code for you. So you can see I've run this code. So I'll just scroll down here past the output. You can see I need a user-specific security token, which is provided here on the page. And then I simply need to paste my query ID, and I'm able to export the data. So you can see I have my data frame where each row is a participant and each column is a variable that I filtered on. And so given that, I'm ready to start analyzing my data and going through that process. So that's a quick tutorial on how to use Picture Open Access to discover data, Picture Authorized Access to select data and export it into a 7bridges workspace. And so I'll quickly pause to see if there are any questions or comments. All right, well, that being said, if you have any questions, happy to stick around and answer them at the end. But for now, I'll pass it over to Dan to go through finding data in Gen 3.
Thanks. Yeah, it was that's really great. Seeing picture presented that way, and that was really nice to see. It's helpful even for me since I haven't really dived deep into what's in picture. Well, my name's Dan, and I work at the University of Chicago in the Center for Translational Data Science as a support analyst. I work in the user services department and work really closely and predominantly on biodata catalysts in the Gen 3 side of BDC. So if you want to go to the next slide, I'll just quickly overview what I'm going to cover. First, I think overviewing biodata catalyst, this ecosystem, and Gen 3 is part of that. Then a couple of different UIs that Gen 3 has for finding data, and then lastly, and probably quickly, how we are also able to hand off data from Gen 3 elsewhere in the ecosystem. So if you want to hit the next slide, excuse me. So I like this diagram. This helped me a lot when I was getting used to this this kind of work, and where Gen 3 really plays their role is a data indexer or a platform for building data commons and data ecosystems, and then being able to efficiently serve those to other parts of the ecosystem. So you can see here, we've got dbGaP. We're grabbing and receiving the authorization for the data and indexing then with these are indexing two data files in cloud buckets, so AWS or Google. We will package those up and then ship them out to well, like we just saw from Emily Picture. We've also got Terra and 7bridges, and actually there should be arrows between Terra and Picture and 7bridges because we're all part of this ecosystem, and there is a lot of interoperability between the different platforms. So yeah, I think we'd hit next slide. Like I was saying about indexing, it's important to think of Gen 3 as not just a a traditional database where we're storing lots and lots of information, in a relational sense. And you're going to be able to come to Gen 3, download your data from Gen 3 and move it on throughout your workflows. It exists out in the cloud, and we're indexing. We do that through giving these globally unique IDs. We call them GUIDs, just colloquially amongst each other. But really, those serve as pointers to the data as it exists, and then lacing in and folding in, authorization, and then interoperability with other areas in the ecosystem is really kind of what Gen 3 is. Therefore, another way to think about how we've structured and how data is structured in Gen 3 is a graph model, where you've got these metadata nodes and then facts or edges between them. So, and it's hierarchical in fashion. So these data that exist can then be linked through several different and very complex levels of metadata, between data files and the clinical information that would apply to them. So I feel like I should pause. I know that that's kind of dense. I know that there's not been questions yet, but I am seeing the chat kind of peeling through. Did we want to pause just for questions or clarifications?
I think there's just a few questions in the chat about accounts and access going on that we're responding to them though. But if
okay,
everybody, yeah, if we want to pause, does anybody have questions so far? All right.
Okay. And then if we want to hit the next slide. Okay, so there are three main ways I think of as far as finding data in Gen 3. I'm only going to be covering one of them really, and just mentioning the other two. We do have a couple of very robust APIs for Gen 3. We do also have a partial GUI hitting a dash query endpoint or suffix onto the Gen 3 Biodata Catalyst URL. But the ones I want to cover are the discovery page and exploration page inside of Gen 3. So I think we hit next slide. It's important to note, if you're ever in, and this is something I kind of learned the hard way and sometimes have to even learn it recently. If you're in Gen 3 Biodata Catalyst and things aren't seeming right or feeling right, there are several features that just aren't available unless you're logged in. So it's easy to have this kind of thing time out or sort of thing. You're, where is this? Why can't I? And you remember to log in. You're like, oh, here we go. So just an important note, and I'm sure it's similar for other parts of the ecosystem as well. Leverage that login and those credentials and get access to all the features. I think next slide.
Looks like we have a hand raised as well.
Oh.
I don't know if you want to address that first.
Sure, yeah. Paige.
Hey, I'm Paige. I use the She series. I am so sorry. From the Biodata Catalyst main website, how would I access this? And you may have covered this already.
How you would access Gen 3?
Yes.
Is there a link within?
If you go under about, I think there's resources and then services. And we have all of the different services and platforms that are on the ecosystem here, so you could launch Terra, 7 Bridges, and then Gen 3 and PIC-SURE also. And they have their respective launch buttons, documentation links, and then some learning links as well. So if you just click any one of those, should send you in the right direction.
Thank you. I appreciate that.
Yeah, it's a great question. The little hack there is that prefix on the URL. So if ever you're feeling lost, also checking that prefix. Where am I? Oh, Gen 3. So that a prefix in the URL is a place. But thank you. That actually I was in the main page a couple days ago looking through, and appreciate you you showing that. Any other questions before I move on? Okay, we can talk about the discovery page. So you'll notice on the Gen 3 page that there are several categories, or they almost look like tabs. They're buttons to these different discovery, exploration, dictionary pages within Gen 3. And right now we're covering discovery. You see in the big orange box, magnifying glass on some study or on some data. This particular search is runs on a microservice we have inside Gen 3. We call it Guppy. It runs on Elasticsearch. It's very fast, very dynamic. It also comes with a couple of additional study filters. So things like if you know which program you'd like to focus on, or you know which type of data you're looking for, you can throw in some study filters. But then that search box here, I've got COPD gene searched, but anything that's associated with these studies is going to be able to pull up very quickly due to Elasticsearch. Yeah, and this is a good way to just start looking at what's in BDC. If you want to go forward one slide, you'll notice that just in the same way that PIC-SURE has variable access to the data, that's true in Gen 3 as well. You'll see these denoted by these little lock or unlock icons. And that doesn't, the discovery page is open, but based on your level of access to the studies, you'll see a lock or unlock. And this is really just essentially how much can you interact with the data. You're still going to be able to see the things in the discovery page, but how much further you're going to be able to interact with that data and dive down into it is really what's in question there with your level of access. And that's again driven by dbGaP. Okay. Let's see, can we hit the next slide? I think we're ready to talk about the exploration page. So the discovery page is quite quick. A little bit more happens in the exploration page. It's located, just next door to the discovery page. But if you want to hit the next slide, one of the first things to note is there are two main tabs at the top: a data tab and a file tab. Really, the difference here is, what are you looking for? You're looking to drill down into specific files that are likely to be in Gen 3, or that you'd like to find in Gen 3, or are there other elements, project, subject, or other variables that you're looking to search on to create a cohort? So we want to hit the next slide. Within the data tab, we've got really just a litany of metadata fields that you're able to filter on. And if you're thinking about building a cohort, you've also got it's much in the same way as PIC-SURE, the ability to get that instantaneous feedback on what would this cohort look like if I was to move forward. So as you filter and create your cohort, you're going to be able to see an instant response in both what is an example of the kind of data you're going to get at the very bottom, as well as the number of subjects, projects, and some other demographics. Next slide. Oh, I think we skipped forward 2. Yeah, okay, so facet filtering. You'll notice in that data tab, there's then 3 facets. And those 3 facets are going to contain and are actually additive upon each other. So this is something that if you're not paying super close attention, could get missed. I think it's pretty cleverly designed, though, that as you're adding filters from, say, the project facet, if you go over to the subject or harmonize variables, those will persist. Those filters will persist. And you do get like a little tiny underline to show, oh, and kind of remind, oh, there are other filters that you've selected that are being applied to the cohort you're generating. So with project, these are, you know, specifically defined pieces of work that you'd be able to think of. This is where I'd like to start building a cohort from, or from the subject level, what kind of, you know, variables you'd be able to pull, you know, a subject level, or harmonized variables. This, you know, very health-centered variables in which you'd be able to pull and push, create your filters. Good. Next slide. So here, if we were to just run through an example of building a cohort using these three facets, if let's say we knew we wanted to use the parent program, you could select that, and you get feedback instantly. 243,000 subjects out of 39 projects. If you want to hit the next slide. And say we wanted to, under subjects, filter out to only female subjects, then grab one more facet under the harmonized variables, say carotid stenosis. We're going to say we'll pick none. You would get that feedback and say, okay, this is the number of subjects or projects, and essentially then have your feedback. And you can see here in this image that you've got those little red underlines to remind you, yes, you did additively build or additively filter from project and subject to where you currently are. Okay. Questions about that? Because we're, you know, I'll handle, you can see the three dark red buttons and one white button the top of this image. That have to do with handing off data. Do we have questions before on the exploration page before we move on? Okay. Then if we want to do the next slide, this is what we were talking about, or what I was talking about earlier, as far as the ecosystem being able to move between these, you know, these buttons will package up the cohort you have actively selected and move them over either to Terra or 7bridges. And if you grab the next slide, once you arrive in those other places, if you want to do the next slide, thank you. You'll even get options there, I believe, the Terra or 7bridges, and how you want to import that data to do, you know, to follow through the rest of your workflow. All right, and I think I have one more slide, which is really just a reminder, and links to a couple of resources for the APIs, which, you know, in my opinion, I feel comfortable with the command line. It's where I like to, you know, feel good there, and that's often where I think Gen 3 is really shining, is through those APIs. Okay. Any questions on that before Dave's up next? Okay.
Thank you so much for that wonderful presentation about Gen 3. Let me just share my screen. Get started. And so Amber just put up a poll. At least it popped up on my screen. So if you just a couple questions for you about TopMed and what data you're trying to access, that would be helpful for us. Okay, so I think I have it all set up. And feel free to stop me at any time. So my name is Dave Robertson. I represent the Biodata Catalyst powered by 7bridges part of the ecosystem. And thank you, Dan, for that wonderful presentation. about Gen 3, because Gen 3 really is the sort of backbone of all this. It provides the authorization to part and allows us to access the hosted data, which is what we're talking about. So this section is going to be about how you can find the genomic files inside a feature called Data Browser. So, and this sort of will carry on from what Emily was talking about. PIC-SURE does great with phenotypes, and you're building a cohort. Emily showed you how to export the cohort to 7 bridges. And so we want to show you the other side of that. Like, what are you going to do next? So in the short time I have, I just want to have you keep a few key concepts in mind. First of all, in the ecosystem, you can find all genomic files for NHLBI hosted datasets in the various ways we've showed you, and in addition, in the data browser. And they're all hosted by Gen 3, and which means you don't have to move the data around. So that's a key value proposition. Data is, of course, very diverse. So we have heart, lung, blood, sleep, as well as COVID data. And there's many open training data sets as well. So for this section, I want everyone to understand how to find and filter hosted study files using the data browser feature specifically, how to link those hosted files to your projects, and understand which are open and which are controlled data sets. So what are some controlled data sets of interest? So of course, the top bed, which Emily talked about. So you have heart disease, lung disease, blood, and sleep. And we have files from Freeze 5, Freeze 8, Freeze 9. So these are really popular. Of course, they're controlled. You have to have downloader status in your dbGaP account. And we can help you sort of get that by working with your PI. So please reach out if you need help with that. And we have COVID-19 data, which is also controlled. So there's several COVID-19 data sets as well. So what are some of the open data? So this is also very popular. Not all the data is controlled. So as soon as you make an account in the ecosystem, you'll have access to 2 major studies. The BioLink training files. So these are training sets created by NIH. They're not real data. My understanding is that they're synthesized data sets for Framingham, Camp, and Digitalis. Those are great for training and for teaching. We also have lots of 1,000 genomes data sets that's hosted by NHLBI. These could be hosted other places, but they're very easy to access on this ecosystem. It includes greater than 6,000 whole genome sequencing files, including the genotype called variants from over 3,000 subjects. Okay, so the feature we're going to focus on today is the data browser. So you get to the data browser, and I'll do a live demo. You get to the data browser after logging into Biodata Catalyst powered by 7 bridges, and it allows you to see all of these hosted studies, either open or controlled, and you can search across multiple studies, and you can use this visual interface to query. It's just a different, it has some of the other same functionality as Gen 3, but it's just a different way to query the data. Of course, the ecosystem is going to programmatically check to make sure you have the appropriate dbGaP approvals to access those controlled files, and I'll talk about that some more. So here's a zoomed in image of the data browser. And now the query we are going to do is rather simple. It's not even going to be this complex. But the idea is you have these boxes, and they represent different entities or different types of data. So you can have subject level data, sample level data, and then finally the output files that you want. So in this example, we have males that are labeled as affected. We want the DNA, the data from DNA as opposed to RNA, for example. And then we want all the CRAMs and BCFs associated with these things. So if you look at the bottom down here, I wish I had a way to highlight this, but down here, you can see the outputs of our query. So there's 19 subjects, 19 samples, and 38 files. So 1 CRAM and 1 VCF per subject and sample. When you get the slides, we have this link down here that will walk you through every button on the data browser. Okay, so for the demonstration, I'm going to try to get through a few of these. If not, please reach out to us and we could just do another demo for you. But I want to show you quickly how to query the 1000 genomes data set and then query Framingham Heart Study for chromosome-based multi-sample VCFs. That's filtered by filter to a specific consent group. And finally, find the genomic. This is actually really popular. We may need to discuss this further as sort of a community. A lot of people are doing or looking at multiple studies, and they want the genomic data structure files already created for them. And I'll show you how to get those if you have the appropriate access level. Okay, so I'm going to flip over here. So I'm just in my web browser here, and I'm just showing you, part of the web browser in Zoom. Okay, so I'm on my home page here, and remember, Emily showed us this project, which we have, Picture and Data Browser Demo. And if I go to the Files tab, it's, well, I made us a folder here, GDS file. So you can make folders and organize stuff like that. But I'm going to grab GDS files from various places and show you how to do that. So why don't I make a new folder? I'll call this 1000. Actually, I'm not going to do that. Let's call this FHS files. VCF, is that what I'm grabbing? All right. And I think I already have, no, I don't. Okay. So I have these two folders, and these are the two control data sets we're going to get. Then I'll show you how to access the open data. So remember, this project contains the phenotype data that was generated by Emily in this Jupyter Lab environment here. So first thing is to use Picture, get the phenotypes, get your cohort, and then go to Data Browser. So how do you get to Data Browser? You go to the drop-down menu and you under Data, and you click Data Browser. And it opens up this new query screen. Now you can always, you can approach this a couple ways, and this is, it is really powerful, and there's a bit of a learning curve, so don't, it takes a little bit of practice. But you can query across these disease types, heart, lung, blood, sleep, as well as COVID-19, as well as the other. So some of these are controlled and some of these are open. And it'll show you at the file level what files are open and what files are controlled. But I'll just tell you that the BioLink data is open, as well as the 1000 genomes project is open under other. Everything else, to my knowledge, is controlled, which means to get access to that, you have to log on with the Area Commons ID, and then the system is going to programmatically check your credentials at the file level. So let's just show you open data very quickly. So if I click on 1000 genomes and I click on Explore One Selected, it'll say, well, how do you want to start? What entity do you want to start with? For these really simple demos, let's just go straight to the files. You could build out a more complex graphical query, but for this community, I've found that it's best to just start with the files. And it shows you that we have 6,000 files total. So what we're going to do is simply search for a specific CRAM format and hit refresh on the file. And so this is going to break it down to 3,000. And now remember, all this data is hosted, so I don't need to worry about storage costs or anything. I can just go ahead and copy all those CRAM files. So these are all the 3,000 plus CRAM files for 1000 genomes. I'm going to copy it to a project I made called Workflow Optimization. And it's not actually moving the data, it's just copying links to the data. And you see that happen really fast. So I'll just open this in a new tab. I'm in a different tab now. And you'll see these are all links. Now, something important, one of our goals is to understand that this is an open, so this is a non-controlled project. There's no message that this is controlled up here by the title. And all of these have an unlocked icon. So that means you can invite anyone in the ecosystem to collaborate on these files. that's why I called the project Workflow Optimization Project. Okay, and so now let's flip back to the data browser. So I just went back to a different the tab I had open. And I'm going to make a new query. I'm going to skip over our second demo, and I'm going to go straight to the GDS files and add those to the controlled project. So I'm still in the new query screen, and it says I'm querying 1,000 genomes. Two things. One, I could save this if I wanted to, or open a saved query that I had before. So if you make something really complex here, you can always save it and go back to it later. What I'm going to do now is change this from the 1,000 genomes open data set to the controlled TopMed combined exchange area files. And it says the current query is going to be lost clear anyway. So let's start with file. So the file entity, and this is saying there's 170,000 files that are available in this database, in this section, which is the combined exchange area. And you'll see that these are all have the red lock icon. This means anyone can develop this query and browse the data, but as soon as I try to copy files to my project, that's when it's going to check my credentials and make sure I have, on a file by file basis, make sure I have the appropriate access level. So I'm simply going to look for something called data format, I believe, yes. So if I click on data format, these are really file extensions. So there's docs, there's BCF, VCF, RData. GDS is a jumping off point for a lot of the R bioconductor-based GWAS workflows. So I want to find those and combine them with the picture phenotypes. So I clicked on GDS and hit refresh, and now I have 46 files. So why 46? They're by chromosome, but if we look closely at the file name, we'll see that these are separated by a minimum depth of 10 or a minimum depth of 0, or essentially 1, I think. So we have the option of picking either of those, or we can copy both. So I'm going to click copy files to project. And remember, this is not actually moving the data, so it happens really fast. The key point is that, and I can tag it with information. I can say these are GDS files. The key point is it's not actually moving any data. It's just copying the links of the data that's being hosted by NHLBI. So if I click on go back to project, and in the files tab, I'll see these. I can then start to organize these a little bit better. I can move these to a better folder, GDS files from Exchange area. And there you go. And then you have your files here, and then you'll be ready to work on the next step, which is finding an app in the public apps gallery. Okay, any questions? Oh, so if there are no questions, I just have some key takeaways for you. I know we're just about time, but use the data browser if you're on 7 Bridges already. Red lock icon means that these are controlled. You need to log in with the area commons, and you need to have the appropriate file level permissions. Although you can make the query in data browser, it won't let you copy it though. The green open files are open to anyone. And you have to have a controlled project, which is another guardrail. So it's only people with controlled access can create or be added to a controlled project.